Researchers worldwide leading the way in biotechnology to promote a healthier and better life.
博奥龙是一家由留学归国人员创办的生命科学领域高新技术企业，专业从事新型生化试剂的研发和销售。公司理念是通过自主创新的品牌产品改变传统实验模式，使生命科学研究变得更轻松、更高效。博奥龙 BioShuttle 系列 RNA 转染试剂，助力您的小核酸药物研发。BioShuttle 系列的 RNA 转染试剂是一种新型的阳离子聚合物，主要适合的样本类型是小于200 NT 的 RNA 分子，如 sncRNA 和 miRNA， 具有转染效率高、细胞毒性小、生物相容性好等诸多优点。该系列的 RNA 转染试剂目前主要有2个货号。KX011049 BioShuttle sIRNA-miRNA 体外转染试剂，可协助将 sIRNA、miRNA递送至真核细胞内。KX011050 BioShuttle sIRNA-miRNA 体内转染试剂，主要用于将 sIRNA、miRNA 通过注射等方式递送至动物体内。通过 knockdown 实验，我们在测试体外转染试剂转染效果的同时，还与市面上常见的转染试剂进行了对比。在过表达 GFP 的293细胞中，使用我们的转染试剂后，能明显地观察到悄降的效果，而且没有出现细胞死亡的现象。BioShuttle sIRNA-miRNA 体外转染试剂适合多种细胞类型。将转染试剂和 miRNA 混合形成纳米复合物，并注射递送到小鼠体内后，通过实时荧光定量 PCR 检测肺组织中 miRNA 的表达水平。结果显示，BioShuttle sIRNA-miRNA 体内转递送 miRNA 的效率极高，且对小鼠无毒害作用。
詹姆斯·沃森说：“生命远远不止 DNA 序列，以此宣布后人类基因组时代的来临。表观遗传学研究正处于飞速发展期，越来越多的科学家开始探讨表观遗传调控层面对正常和疾病状态下生命活动的影响。广州表观生物科技有限公司应运而生。”表观生物创立于2016年11月，专注医学表观遗传与转录调控研究领域，提供尖端的新一代测序技术服务和试剂盒开发，为核酸精准诊断与治疗靶点开发提供先进的解决方案，服务国家健康中国发展战略。通过自主研发，表观生物已经建成国际领先的表观基因组、表观转录组、转录与调控、翻译组、单细胞组、胞外囊泡 RNA 等多个组学测序技术，并搭建表观云自动化生物信息分析平台，已推出了多项市场首发服务项目，包括超级增强子鉴定 High-Trip-seq、超微量 Mirib-seq、Slam-seq、Grow-seq、PANDORA-seq 等高端科技服务项目。表观生物配备自动化一页工作站，可用于核酸提取纯化、测序文库构建、高通量药物筛选。表观生物经过多年研发，自主搭建和外部引进多个单细胞测序平台，包括液滴法单细胞平台、液滴融合单细胞平台、微孔法单细胞建库平台。表观生物至今服务超过140家高校科研单位及3级甲等医院， 100家企业，客户研究成果发表于 Nature Methods, Gastroenterology, Circulation, Blood 等知名刊物，并与多个科研单位建立产学研合作关系，先后获得中科院发表于 Nature 杂志的 RNA 与 RNA 互作 RNA-seq 技术，以及北京大学 Select M6A 修饰定量技术的转化授权。在2018到2021年，表观生物成功举办了5期表观专题研习班和2期医学表观遗传学前沿技术培训班，邀请众多本领域知名专家嘉宾，受到广大学员的热烈好评。2019年，表观生物获得国家高新技术企业认定，并在2019年与2020年2度获得广州市黄埔区邓明培育企业认定。2021年入选广州市专精特新企业。表观生物以促进医学表观遗传学研究的深入发展和临床转化为使命，愿与医学科研工作者及医药研发机构一道，协力破解重大疾病发生发展的表观遗传分子机制，积极推动以表观组学为基础的精准诊断以精准治疗的转化医学应用，造福人类健康。
sncRNA is the pioneering DNA RNA synthesis company known for its innovative R&D capabilities and commitment to continuous improvement in synthetic instruments, raw materials, and automated production systems. Our state-of-the-art integrated platforms empower us to deliver cutting-edge synthetic genomics products, unlocking the potential of genetic research. From the successful development of the first-generation synthesis column in 2009 to the introduction of the first oligosynthesizer in 2014, and finally the establishment of an independent factory-style service model covering the entire nucleic acid synthesis industrial chain in 2020, sncRNA has accumulated rich synthesis experience and stable supply capacity. As of 2023, the company has provided gene synthesis, modification synthesis, antibody and proteins, gene regulation, gene sequencing, chemical reagents, molecular biology reagents, equipment, and a variety of technical services to over 200,000 customers worldwide. In the future, sncRNA will collaborate with researchers worldwide, leading the way in biotechnology to promote a healthier and
Oh, hi, Ryan.
Good evening. How's it going?
Yeah, good morning for you. Good evening
Yes,
for us.
yes.
Yeah, we are great. Yeah, the time is just right. We just start just at 8:30, so we still can
Sure.
chat a bit. And I
Yeah,
see Yishuan is also here.
I think so.
Yeah. I also even see some people from UK, you see.
Oh, nice.
Yeah. Why Yishuan? Yeah, I just chat with Yishuan because you just show up at the right time.
There's Hey, X, how's it going?
Good. A lot of people.
So Yishuan is in the same city as we. We are all in Guangzhou, south part of China.
Yeah.
Very close to Hong Kong.
Got it. I need to get better at my geography, but I got to visit China like when I was very young, actually. It was very fun, but I didn't go to the southern part. I was in Beijing and Shanghai and Xi'an, actually. But so I think I saw all the touristy places.
Yeah. The
But
best city you already been there since Xi'an.
but it was
so, yeah.
yeah, but it was in 2007. So it was like, I mean, I
Oh,
think the cities are quite different now. They're much bigger, and um, the scope of everything is very different. So I think it'd be fun to visit again
yeah. That's a
sometime.
big change. That's more than 15 years, right?
Yeah.
Okay, seems the time is right. So I will give an introduction for you, then let you start.
Recording in progress.
Okay, it's great honor for us to have Ryan Flynn from Harvard University to give us a lecture today. And Ryan received his bachelor degree in biology from MIT. It's from 2006 and 2010. And then completed his MD and PhD in cancer biology at Stanford University from 2010 to 2017. And after that, he did his postdoc training at the Stanford University Department of Chemistry with Caroline Betrotti, right? Who just got the Nobel Prize 2 years ago. That's 2022, which is for click chemistry. And Ryan, of course, when he as a postdoc at the Caroline's lab, who did a very important discovery for glycoRNA. So today he will talk about his finding and some new things in his lab just happened. So let's welcome Ryan.
Awesome. Yeah. Thanks everyone for staying up late. I might even be already asleep by this time in the night. So I appreciate everyone coming. So I'll just share my slides here. Today I kind of want to go through a bit about the ideas of glycoRNA. And I think my goal for today is to kind of paint this picture of glycoRNA biology on the cell surface. And so I think that what hopefully will become apparent is that, we're in the very early stage of, I think, an exciting new field. And, there's a lot of tools that have to be developed, a lot of biology to be discovered that could be, I think, very exciting. But it's sort of a new way to look at biology and how cells might operate. So, of course, part of this name, I think, hopefully people can see my mouse here. Oh, maybe.
Yeah. You can also change the laser.
Let's see. Now you can see Spotlight, maybe even easier.
Yes.
Okay. So, part of this beginning, I'll talk first about the glyco part, then some RNA, and then on the cell surface. So it's sort of in the order of the title. So when we think about glycobiology, this is sort of a distilled version of what someone might think about on the mammalian cell surface. So we have our plasma membrane here. This is the extracellular space. And while I won't get into the details of the carbohydrate chemistry, you can see one example of a glycan, of a monosaccharide here on the left-hand side, and it's called sialic acid. So it's a medium complicated structure, but we cartoon them with these colored shapes to simplify the illustrations. And what I want people to take away from this slide is that there are a lot of colors, there's a lot of shapes, and the organization is very diverse. So we put glycans on proteins, we put glycans on lipids, and we make very complex architectures of lipid glycan protein hybrids called GPI anchors. And so this diversity of carbohydrate polymers and carbohydrate conjugates is sort of paired with the importance of this in biology. And so when we think about mammals, especially like humans and mice and everything, glycans do a ton of really important things. They will control the shape of a membrane. They'll control how cells talk to each other. We think a lot about how bacteria and viruses leverage or hijack glycans and carbohydrates and how this is part of the pathogenesis mechanisms. And just even within our own bodies, there are major paradigms for how the immune system communicates through carbohydrates. And so the immune recognition and regulation is really controlled by glycans. So I just want to say that glycans are really exciting and they're very important to study. But I mentioned that this is even a simplified version of what the cell surface might look like, because really it's a very complex landscape. This is even more zoomed out a bit, and this is the membrane down here. And we're looking at glycolipids and GPI anchored proteins here, these things called proteoglycans, which could have heparin sulfate or chondroitin sulfate. We have regular N and O glycosylated proteins. But my training had been traditionally as an RNA biochemist. And what you'll notice here is that every other biopolymer is included, but not nucleic acids. So when I was a postdoc, I wanted to ask this question: why is RNA missing? I thought that either the cell definitely excludes RNA because it's bad to be outside, or people hadn't asked the question, right? We were just missing it. And so more recently, people have been illustrating the surface of the cell. This is from a review from this year. And very interestingly, they now cartoon glycoRNA into the illustrations. But you'll notice, and probably fairly, there's a question mark here. There's still many open questions about this. So while people are getting confidence, a little bit more confidence and excitement about the idea, there's still a lot of curiosity and questions and thinking. So let me tell you very briefly what we originally reported about discovering the glycoRNA, and then I'll tell you about some of the new work and the new tools that we've been developing to study them. So initially, what we did was we used a strategy leveraging substituted sugars, metabolic reporters, and cell culture. And so this is actually what my postdoc advisor had won her Nobel Prize developing. So this is sialic acid with an azide substitution, which allows for click derivatization. And you can do this on live cells, you can do this in vitro. But for the past 2 decades, people had been only isolating proteins or only isolating lipids when they labeled with these glycan reporters. So I decided to extract the RNA and ask, is there ever a glycan reporter in the RNA fraction of cells? And we could use a very simple, like northern blot gel readout to first determine what was going on. So if we saw biotin, which was conjugated to our glycan reporter, we could infer that there was glycans with the RNA. So what does this look like? Just for this first example, I'll show you what a sucrose gradient looks like. And this is I think, a very satisfying biochemical result, where if you fractionate by density, you will see very small RNAs on the left and very long RNAs on the right. And this is what the total RNA would look like in a single lane. So you have your ribosomal bands and the small RNA. And what we found was that the biotin signal was very slowly migrating over here in the total sample. But when fractionated for the RNA transcripts, completely fractionated with the small RNA. So this was telling us that these glycoRNAs were being modified on small non-coding RNAs. And when we went to do sequencing, this is the last from the published work, the original published work, what we found was that there was a very specific class of small non-coding RNAs that were enriched with the glycan. So we did sequencing in 2 cell types, human ES cells on the x-axis and HeLa cells on the y-axis. And you can see that there's a distinct class of RNAs that are enriched in both. But what was very interesting was that almost every RNA that was enriched in both. And so it seemed like the types and the specific RNAs that were glycosylated were common across these cell types. And so it led us to a very original model that was very, I would say, cartoonish, very rough to start, but it made us think that on the surface of the cell, there were glycoproteins, glycolipids, and now glycoRNAs. So where do we go from here, and how do we think about this now? So this is a fine illustration for what could be on the surface of the cell, but there's a lot that we don't know. So the first key problem and the biggest question that we would get asked, which was fair, was what is the molecular connection between an RNA and a glycan? We had indirect evidence that glycoRNAs existed, but what was the direct evidence of that? The next thing was really, what are all the types of glycans that are on the RNA? I'm illustrating here a very complex N-linked structure, but is this a real structure? Is this the only structure? We don't know. And it's very important to know what types of glycans are on the RNA, because what the RNA could be doing could be dictated by the glycoform, the particular glycan structure, because there's very specific receptors that will bind each type of glycan. So if you have one type of glycan, you might be doing one thing, and if you have another type of glycan, you might be doing another thing. So the scope of glycan modifications is actually super important. And the last thing that I'll describe is what we've been discovering about the context in which the glycoRNAs are actually organized on the cell surface. So here we're sort of illustrating that the glycoRNAs are floating above the membrane, perhaps with no tether, but this is not actually what we think is going on. We think that there is an anchor, probably a protein, that is holding the glycoRNAs here. So I'll first start to describe the evidence we've generated about the linker, because if you don't think a glycoRNA is a single molecule, the rest of the talk is not very interesting. So how do we go about doing this? So I mentioned that originally we used a azido sugar. So this is an example of sialic acid with an azide incorporated in it. And the convenient thing about the azide is that you can do click chemistry. So here at the bottom is an illustration of a copper-free click reaction that has occurred that can install biotin on the sialic acid for detection. Now, this is very nice, but the problem with this metabolic reporter strategy is that the cells need to be alive. The cells need to be able to take the MAN-NAS, which is the precursor to this, into them and actually metabolize it. And the sial transferases that add sialic acid to the glycans need to be able to actually incorporate it into their pathway. And there's a lot of limitations with these substituted sugars. So what we did was we actually developed a tool that could derivatize sialic acid without any metabolic incorporation. So we looked at the sialic acid and said, well, there's this really interesting, very reactive chemical region here, which is a cis or a vicinal diol, which could be oxidized. And so what we did was we did very mild oxidation, which can break this bond. It leaves an aldehyde residue after the oxidation, which can then be ligated with something like an amino oxy. And in this case, we can also install a biotin for detection and pull downs and these sorts of things. So this would have let us look at glycoRNAs from any sample without metabolic reporter incorporation. So we could go to a patient sample, we could go to a mouse sample, we could go anywhere we wanted and look at glycoRNAs. So what does this actual detection strategy look like? This is an example. I showed you previously what the glycoRNAs look like, and we know the glycoRNAs run up here in this high molecular weight region. So this is detecting the glycoRNA with this rPAL signal. And what you'll notice is that all of the biotin signal goes away with RNase, but only this high molecular weight region goes away when you add sialidase. So what this was telling us was that our labeling was very good and very selective in this high molecular weight region for the glycoRNA. Now, people will notice that there's some signal here at the bottom that is RNA dependent, but not glycan dependent. And this is because the ribose in RNA has vicinal diols, has a 2', 3' hydroxyl. So we think this bottom signal here is background labeling of the RNA, while the top band here is very specific for the glycoRNA. But still, in a gel, this allows us to very confidently and specifically detect glycoRNA in this high molecular region. So all of this was done in vitro to demonstrate the specificity and the efficiency of the labeling. But I mentioned that we found the glycoRNAs to be on the cell surface. So could we prove that this method still detects the surface glycoRNA? So the last quality control that we did with this method was to take cells, unlabeled cells, and we added the sialidase enzyme while the cell was alive to cut all of the sialic acids off of the surface of the cell. And then we washed the cells, then we extracted the RNA, and we processed for this glycoRNA labeling. And what you'll notice is that in as little as 15 minutes, we get a very strong reduction in this high molecular region. But after 60 minutes, we get almost complete reduction of the glycoRNA band, which suggests that almost all, if not all, of the signal that we see with this labeling technology is reporting on the surface presented glycoRNAs. And again, this bottom band and these bottom bands here are not affected by the sialidase, either in vitro on the left or on cells on the right. So all of this was saying that we now have a tool that can label any glycoRNA and perhaps all of the glycoRNAs in our sample. The critical point here is that, and I'm not showing the quantitation, is that this labeling technology is between 10 and 50 times more sensitive than the metabolic reporter. So we can really see more glycoRNAs for every amount of RNA input that we start with. So what do we want to do with this? I mentioned we want to show, and we wanted to try to figure out the linkage between the RNA and the glycan. So I turned to my now good friend, Ax, and worked very diligently with him to figure out a way to look for the connection between the RNA and the glycan. And the way that we developed this strategy was the following. We took a lot of RNA to start. We started with tens of milligrams of total RNA. We purified that down to single-digit milligrams of small RNA, after which we did the rPAL labeling. And then we could pull down with neutroavidin beads to remove all the non-glycosylated RNAs. And the first thing that we did was we removed all of the uninteresting peripheral nucleosides around what we thought was this very important key glyconucleoside in pink here. So what I want to show you first is what RNA were we actually enriching with the glycan? So what were all these peripheral nucleosides on the glycoRNAs that we saw? So when we looked at the nucleosides that were eluted off of the glycoRNAs, what we saw was that if you rPAL labeled, so in the second and fourth lanes here, you saw a really strong enrichment of a huge diversity of modified nucleosides, base modifications, ribose modifications, highly modified nucleosides. And what this was telling us was that the pool of RNA that we enrich with the glycan are very heavily modified. This was in line with the fact that when we did the sequencing, we saw many tRNA and small non-coding RNAs, which are known to be heavily modified. So what we saw from this was that, in fact, we were getting back the expected RNAs, and we could see those PTMs that were coming down with the RNAs around the glycan modification. But like I said, these are the boring, non-glycosylated nucleosides. So how could we process this material now to identify what was this nucleoside and what was the linkage that was connecting it to this glycan? Well, it turns out that in glycobiology, there's a really interesting enzyme called PNGSF. PNGSF is an amidase, and it will cut the bond between the template that's N-glycosylated and the first sugar, the GlycoRNA. And that's cartooned here in a blue square. So what's very interesting about PNGSF is that when cutting, it will release a carboxylic acid scar. So normally this results in a transition in amino acid identity from an asparagine to an aspartic acid. So if you're a proteomics person, you can search for aspartic acid containing peptides that should be an asparagine, and you can determine which peptides were glycosylated through this transition between asparagine and aspartic acid. So we thought that maybe a similar thing could happen with the N-glycans. Maybe if we saw nucleosides come off of our beads after PNGSF treatment that had a carboxylic acid, that could also be a scar from the glycan modification. So what did we do? We set up a number of digestions. These are very large-scale experiments because we need a lot of material to be very confident and have enough sensitivity in the mass spectrometer. So we did this from 2 different cell lines, and we did it in different orders of operations. We would either do the digestions on the beads, or we would release from the beads and then do the digestions in solution to make sure that we weren't seeing any artifacts of the way we were doing the digestion. And what this plot is showing us is the number of nucleosides on the y-axis that are intersecting with each of these data sets. So this would be a nucleoside we found once in these 2 data sets, so not very interesting. What we really want to focus on are the nucleosides over here that were found in every single data set that we generated, right? These are commonly found after PNGSF elution. The most interesting one is this AC3PU, which looks like this. Now you'll notice you have a ribose, a uracil, and then this linker structure, which terminates in a carboxylic acid. So this is perfectly in line with our hypothesis that AC3PU could be released due to cutting of PNGSF. Now I want to bring up something else that we reported in this work, but we didn't follow up. So if there are any chemists and nucleoside people that want to think about this, it's a very exciting thing to consider. We also noticed in this list, YW86, which is Wibutacine 86, and also here, YW72. These are the structures of Wibutacine. And what you'll notice is that these are hypermodified Gs that also terminate in a carboxylic acid, both of them. So of the top 4 nucleosides, 3 of them terminate in carboxylic acids. So we think this is a very rich data set, and I'll show you our validation of AC3PU in a few slides, but Wibutacine we think is a very exciting opportunity to think about other linker structures that are connecting RNA to N-glycans. But I'm not a good enough synthetic chemist to generate Wibutacines, and so we need more help to think through proving these other linker structures. So again, I won't talk more about the Wibutacines because we didn't do more work on them, but AC3PU was very achievable. This was something that we thought we could actually measure. And we wanted to go a little bit deeper to try to prove that the structure that we were seeing coming off the RNA was really due to the PNGSF cleavage. And the way that we would prove this would be the following. We could take our material, just like I showed before. We could sialidase treat it to release it from the beads, which now we have things in the solution, in the reaction mixture off the bead substrate. And what we could do is perform the PNGSF cleavage either in normal reaction with normal water or with heavy water containing oxygen 18. And the reason why this is interesting is that when PNGSF performs its cleavage, it uses the water in solution and then takes that oxygen to install it in that carboxylic acid scar. So if the PNGSF was doing the job of releasing AC3PU, if we digest it with heavy water, we should see heavy oxygen in the resulting metabolite that we see in the mass spectrometer. This would confirm that the ACE3PU is coming from our digestion and not from a cell itself. It was coming actually from the cleavage after P and JSF. Now, to show you what these look like from a chemical perspective and from the masses we expect, what we're seeing here is that the full ACE3PU would be 346. This is the 2 Dalton shift. And in the instrument, we'll end up seeing a ribose loss in the MS2, which would result in a mass of 214 or a 2 Dalton shift here at 216. So when we evaluated what we saw in the instrument, now looking at a synthetic standard, which is very important to compare to when you're looking at novel molecules in a mass spectrometer, and we compare the elution profile to what we get when we do this digestion with light water or heavy water, we see that there's a peak that elutes at exactly the same time in the LC. And when we look at the MS2 fragments that we see from this injection time, what's very exciting is that we saw a very consistent and exact mass for our standard. We saw the same parent mass for the light, and we saw this 2 Dalton shift from the heavy. And they all fragmented the expected ribose loss of 214, 214, or 216. So this is very exciting because it was the first evidence that we think there could be a glycan connection to the RNA through ACE3PU. But this is sort of the shadow or the scar of a linker. It's not really the linker itself. So how could we try to find the linker structure in the instrument directly? Well, what we could do is we could go back to our glyco toolbox. We could use different enzymes now that cut further away from the linker structure. So what we did was we used these enzymes called EndoF2 and EndoF3, which cut one more unit away from the linker. So this now would include the ribose, the uracil, the linker, as well as the GlycNAc. So what we're calling AC3PU GlycNAc. And that would have a mass of 548.2. And so what we did was we synthesized this synthetically. And again, we compared what our synthetic standard looked like compared to what we could elute off of an RNA from an RNA sample. And again, when we looked at the elution profile from our expected mass in the instrument, we could see that there was a very similar peak between the synthetic standard and what we got from the RNA. And very excitingly, when we look at the fragments in the mass spectrometer, we could see that the parent mass was our expected size. We could see the loss of the GlycNAc from this molecule, as well as the loss of the ribose down to this 213 structure. So this was now, we think, the first evidence, direct evidence in a mass spectrometer that there was a direct linker between the uracil and the N-glycan GlycNAc. But what's actually doing this in a cell? What is producing this linker structure that would allow N-glycosylation on the RNA? Well, it turns out that a few years ago, the enzyme family that produces AC3PU was discovered. And this is the DTWD family. And it will take a uracil with a SAM intermediate and produce AC3PU. And it had been described that there are two major enzymes that do this, especially on tRNAs, where one of them is more restricted and the other one, DTWD2, is active on all of the tRNAs, as well as is more broadly expressed across all tissues. So we thought that we should try to define if one of these enzymes was responsible or could play a role for glycoRNA biogenesis. So we focused on DTWD2 because it was very broadly acting in the literature and broadly expressed. So what we found after we knocked out DTWD2, these are two different clones of DTWD2 knockouts, is that there was a very specific but minor loss of glycoRNA signal. You'll notice this quantification here is about 10% loss from the wild type. So there's a distinct loss, but it's minor. What was very interesting, though, was if you look at the molecular migration of the glycoRNA, after you knocked out DTWD2, there was a clear shift. And if you look at the gel down, all the glycoRNAs ran further into the gel when you knocked out DTWD2. So this is very interesting because we saw both an intensity difference and a molecular weight difference. And the molecular weight differences we ascribe to glycoform changes in the glycoRNA. Now, just to sort of close the loop here, what's very interesting is that deletion of DTWD2 is sort of purely an RNA enzyme change. We're only changing ACE3PU, and we're seeing an effect on the glycoRNA. Now, what happens if we do this on the glyco side? We see the same effect. So this is the same data showing you the DTWD2 knockout and the shift down. If you look at the effect of PNGSF, we see the exact same effect. So treatment of the glycoRNA with PNGSF shifts the molecular weight down. So if we genetically alter the RNA substrate through DTWD2, we see a downshift. And if we enzymatically alter the glycans on the RNA with PNGSF, we also see a downshift. So we think this provides really coherent evidence that there is a direct connection through an N-glycan to the RNA via ACE3PU. And the molecular model that we've built out here is illustrated in this slide, where we start with a naked tRNA that, through the work of DTWD2, will be modified with ACE3PU, this first linker. And we think that there is an initial step where this modification gets amidated and then could lead to, for example, translocation of this molecule into the ER. It could then get N-glycosylated with the primary oligosacchαρyltransferase, and then eventually moved to the surface of the cell after elaboration and sialylation to be presented in its full form on the surface of the cell. So we've been building out this molecular model and now have a better understanding of this sort of covalent connection between a uracil and an N-glycan. But when we zoom back out, the question really becomes, is this the biggest scope? Is this the real full nature of all of the glycoRNA glycans? What is really modifying them? So what I'll tell you is that it's actually a very narrow scope to only think about N-glycosylation. If you're a glycobiologist, you'll know that there are very major classes of glycans, including N-glycans here, which start in the ER. They get made through the work of OST, trafficked into the Golgi, trimmed and elaborated first with NGAT1 and then through a bunch of other enzymes in the Golgi. But there's this parallel process of O-glycosylation, which happens almost exclusively in the Golgi, starts with O-galnacylation. It then often will be added a galactose here, and then further elaborated with some branching structures and sialylation later in the Golgi. But all of our work to date had been really focused on N-glycosylation because of our evidence with PNGSF and now this linker. But our question was, is that the full scope of the glycoRNA glycans? And the answer is no, right? And so the way that we learned this was by doing genetic knockouts. And we focused initially on knocking out OST to look at N-glycosylation. And these two enzymes here, C1-GALT1 and COSMIC, because these are the sort of the funnel point to make most of the mature, branched, and sialylated O-glycans. So what we first looked for was just what happens to our rPAL signal if we get rid of these key enzymes. And this gel, I think, is very revealing and sort of very obvious. If you get rid of one of the two N-glycosyltransferases, not much happens. If you get rid of the other one, not much happens. These are a bit redundant with each other, so there's some possibility that they could be still doing some things. But the data for COSMIC is quite obvious, I would say. And we were very surprised by this. You'll notice that when you get rid of COSMIC, there's almost a 70 or 80% reduction in the total amount of glycoRNA that we generate. So this was our first indication that O-glycans are possibly a really big contributor to the amount of glycoRNA signal that we see. I'll also remind everyone that I just showed you a gel of the effect of DTWD2 knockout. That effect was about 10%. That's an N-glycan. Now, conveniently, if you add 10% and 80%, you're almost at 100%. And so what we thought was very interesting was we were working on these two projects in parallel, and we saw a very specific but minor effect with N-glycans and a very major effect with O-glycans. And then independently, the sum was near 100%. So we think it's all consistent, but it was very surprising to us because we had been very focused on N-glycosylation to start. So what did we do next? Well, we went to the actual glycosyl transferases. So these are actually just the second step in making O-glycans, but these GAL-NT enzymes are the actual glycosyl transferases that generate the O-glycans. So what you can see here is that by deleting 3 of the 20 O-glycosyl transferases, these O-GalNAc transferases, we can reduce the total signal of the glycoRNA by about 60%. So again, there's many O-GalNAc transferases, and we didn't have the ability to knock out all 20. But what this was telling us is that the actual glycosyl transferase activity is also very important for the rPAL signal. The last thing I'll show you here is that we've gone in and actually, again, with the expertise and help of AX, we've gone deep and looked at the actual types of glycans that we see on the RNA. And what we were able to do was use 2 types of mass spectrometry. On the left-hand side, I'm showing you data-dependent acquisition, O-glycomics, and on the right-hand side, I'm showing you what we're calling glycan DIA, or data-independent acquisition, to define the glycoforms that we can see on whatever protein or RNA of interest, but in this case, it's all small non-coding RNAs. And what you'll see is that many of the structures that we have are sialylated O-glycans. And this is showing you signals from K562 cells as well as HEC293 cells. And the same types of glycoforms that we see in the DDA, we also see in the DIA, and they cluster very similarly. Although with the DIA, we see a slightly deeper set of glycoforms because the data type is a little bit, or the experimental strategy is a little bit more sensitive. So what we were very excited to see from this is that there's actually a robust and interesting diversity in O-glycans on the RNA. And so if we come back to the model, we start to see it evolve. Right now, we see these more specific N-glycans. We have our linker here now attached to an RNA. But really importantly, we have O-glycans decorating the RNA, these shorter, highly sialylated O-glycostructures. And so I think this is starting to represent a better model of what these glycoRNAs are actually looking like on the cell surface. So with the last maybe 10 or 15 minutes here, I want to tell you a little bit about this organizational idea, this broader concept about what's going on the cell surface. And the short answer to start is that we think there's a bigger and broader set of RNA biology happening on the cell surface, which is nucleated by RNA binding proteins. So what did we do to actually get here, and why were we asking this question? Well, the question was motivated by thinking, how does this happen? Is the RNA stuck here to the membrane, or is there a protein intermediate? So we made a very simple question. We asked, what proteins are on the cell surface? And to do this, we said, well, many people have asked this question. There are many different experiments where people have produced surface proteomes or surfaceomes. People will use glycan labeling. People will use sulfonyl NHS labeling to get to the cell surface proteins. And we thought, well, everyone has defined already what's on the cell surface. And in the RNA biology world, people have tried to define what binds an RNA. what is the RBPome? And they've also used mass spectrometry techniques. So we thought we could intersect these two data sets computationally and ask what is the intersection, what is the overlap, which would define and probably tell us what could be nucleating and holding the glycoRNAs on to the cell surface. The surprising thing for us was that RBPs shouldn't go to the cell surface. RBPs do not have transmembrane domains, and RBPs do not have signal peptides. So they are not instructed molecularly to get to the cell surface. So we actually thought that there'd be very few RNA binding proteins that are on the cell surface. But in contrast, we actually found that there are hundreds of RNA binding proteins that are overlapping with our surface proteomics data sets. So this would say that there were 200 RNA binding proteins, 209 RNA binding proteins that were found in all three data types that were published in the literature. So this is very surprising to us because there should be no reason why and no mechanism on how they get to the surface of the cell. So what does this actually look like? We were very excited by this and we had to validate this. We had to think about, what does this mean and can we show that it's real? So the way we did this was taking one of our favorite RNA binding proteins called DDX21. DDX21 is a nucleolar helicase. In here in blue is the nucleus, and in yellow is DDX21. That's the signal that we're seeing. So when you would normally stain your cells, you might fix them on a glass slide, permeabilize the cell, add the antibody, and do your imaging. And this is a confocal image. Looks beautiful, looks like exactly what we would expect, just goes to the nucleolus. But in a tissue, in a living organism, when you're an immune cell looking at a cancer cell, you will not permeabilize your partner, your friend that you're talking to. You'll look at the cell surface first. So we asked, what happens if we change the order of operations? What if we add the antibody to the cell while it's alive, before the membrane is permeabilized? And what you'll see is that now the yellow signal is all spotted around the cell surface, completely extracellular. So there are many domains on the outside of the cell that have DDX21 presented while the cell's alive. This is just one example. I'll show you another example here, now reconstructed in 3D. The left-hand side is another RNA binding protein called hnRNPU. The right-hand side is in yellow, beta-2 microglobulin. This is a classical membrane protein. So beta-2 microglobulin is very highly expressed. You can see it coats the entire cell. It's very dense on the surface of the cell. Whereas the RBPs have these more punctate clusters, but they also are on the surface of the cell. They're on the outside, topologically extracellular side of the cell. So we have many evidences that RNA binding proteins are extracellular, presented on a living surface of a cell. But this was a confocal image. Even though it's in 3D, it's still limited by the diffraction of light. And we saw these domains that looked very interesting. So we wanted to zoom in even more. So we set up a super resolution imaging experiment. So using DStorm, which will get us about 30 nanometer resolution, we looked at the same cell. And again, this is for hnRNPU. And if you look here, this is the cytosol. This would be where the cytosol is. This is where the outside of the cell is. And this is a slice of the membrane. And what you'll notice is that now we have very distinct punctate clusters. And when you measure these clusters, they're all about 150 nanometers in diameter. And they have very specific spacing between each other. So they're about 300 nanometers apart from each other and 150 nanometers in size. So this is very interesting because when you compare this to the same cell with a different membrane stain, like beta-2 microglobulin, completely different, right? Again, cytosol, extracellular space, and this is now what you see for beta-2 microglobulin. It's fuzzy. There's no clustering. There's no domains. Beta-2 is just everywhere in the membrane. So this was telling us that there's something special, something unique about the clustering and the organization of the RBPs that was different than other surface proteins on the same cell. But these are still quite large domains. There could have many, even dozens of proteins within each cluster. So what else goes into these clusters with the RBPs? Well, at least with this RBP. So what we started to do was two-color simultaneous imaging for multiple things on the cell surface. So I'll show you one example here with now DDX21 as the yellow color and DNA PKCS, which is another RNA binding protein that we found to be on the cell surface. You'll notice both of them generate this very obvious cell surface pattern. And if we zoom in to one of the junctions between these cells, you'll see many spots that are white. They have overlapping signal. And if you do the quantification, almost half of all the spots are white. So many of the places where there's RNA binding protein A, there's another RNA binding protein right there. They're in the same physical domain. But it's very interesting because this is totally different when you look at the relationship between a classical membrane protein like beta-2 microglobulin and an RNA binding protein. This is the zoomed out view, but in a zoom in, you'll notice that there's no white. There's no white anywhere. So the RBPs are in a distinct place from the classical membrane proteins. They're nowhere near, at least in the confocal dimension, these classical membrane proteins. So this is very interesting because it was telling us that there's a biochemical specificity to the RBPs being near the RBPs. But we got into this question not about RBPs. We got into this question about the glycoRNA. We were thinking glycoRNA. So where's the glycoRNA, right? So now we could go and look at the RNA question. So we have this antibody called 9D5, which binds RNA. And if you stain the surface of the cell with 9D5, you will also get spots on the periphery. This is a co-stain for DDX21, and what we'll notice is if you zoom into a single cell, many of the spots where there is DDX21, there is also RNA. So we could show that in space, the RNA and the RBPs co-localize, but this was just a general RNA stain. So it's not biochemically defined as glycoRNA. So how could we do that? Here we turned to a proximity labeling tool where rather than recruiting a dye to do imaging, a fluorophore to do imaging, we could recruit HRP. And with the HRP, we can spray biotin in proximity. So when we spray biotin in proximity, we can look for what are the RNAs that were labeled with biotin around our antibodies. And what we saw was this very interesting pattern. So if we spray biotin around and anchored around this 9D5 antibody, we could get this smeary signal here on normal cells. If we RNase treat, everything goes away. And if we sialidase treat, the signal, you can see this signal and this signal, they shift up. The bolus of signal now moves up. And what we've defined in other experiments is that when you have a glycoRNA, if you shred and you remove all the sialic acids, now the charge to mass is lower. Its negative charge is lower for the same length of RNA, which means that in a gel, it will run slower. So it will be higher and closer to the well. So this is sort of a diagnostic feature that says this is glycoRNA signal. So putting all this together, now we take our model and we say that there are actually CSRBPs, cell surface RBPs, that are nucleating and clustered around the glycoRNA on the cell surface. So our question with all of these discoveries is, what are they doing? Why would a cell put them on the cell surface? So this is a big and exciting question that we are contemplating a lot. And one set of biological contexts that we've been thinking about this in are other contexts where very negatively charged biopolymers, negative charge, plays a role on the cell surface. And I bring this up because RNA is very negatively charged, but there's this more classical negatively charged biopolymer called heparin sulfate that's widely reported in a bunch of different biological contexts to have activities on the cell surface. And so briefly, heparan sulfate proteoglycans look like these. They have membrane anchors, and then they have these linear chains of negative charge. And the mechanism is that they are usually there on the surface of the cell to bind factors from the serum. And classically, for now decades, people have known that cell-penetrating peptides bind heparan sulfate proteoglycans. And cell-penetrating peptides are very interesting because they have really important roles for drug delivery, cellular targeting, and cell entry, because they're very short, often cationic peptides that can deliver cargo inside of cells. But the mechanism for how CPPs have worked, to be honest, I think is a little unsatisfying. It's not very robust, and there's still a lot of open questions. And for example, the first CPP that was ever discovered, and still probably one of the best ones, is from this protein called TAT. Now, TAT is interesting because TAT is actually an HIV-encoded protein that very specifically in the biology of HIV binds a viral RNA. TAT is specifically designed and specifically evolved to bind this TAR RNA. TAT's a bigger protein, but the TAT CPP comes from this interface, comes from the binding region between the TAR RNA. So we made this interesting hypothesis that perhaps the reason TAT interacts with the surface of the cell is not only because it interacts with HSPGs, heparan sulfates, but because TAT itself is an RNA binding protein, and there's RNA on the surface of the cell. So we thought that perhaps the TAT CPP could have dual modes of binding heparan sulfate and also binding RNA. So we set an experiment up to test this. The really interesting thing about TAT is you need very few amino acids to have its cell binding and cell penetrating activity. So we could take these amino acids, tether them genetically to GFP, and perform a series of experiments. You'll notice this is very cationic, arginines, lysines, everything. It's very cationic. And so what we did was we made a general GFP with no TAT, and we made this TAT GFP fusion. We could add these to cells, and we could ask, do they bind cells? Do they go inside of cells with and without RNA on the surface of the cell? And I'll show you this key experiment that we were very excited about. So if in a normal situation, you take cells and you add GFP to them, these were HeLa cells. You're growing HeLa cells in a culture dish, you add the GFP, no transfection, no nucleofection, just GFP in solution. You will get a little bit of internalization, but you'll notice it's very granular. It goes to the endolysosome. It's stuck in organelle. If you fuse that TAT peptide, that little TAT peptide to the GFP, it's a very dramatic difference. You'll notice more GFP signal, and now the GFP gets into the cytosol, it gets into the nucleus. It goes everywhere in the cell. So this is the classic cell penetrating phenotype. What's very interesting, and we were very excited about, is if you treat the surface of the cell with RNAs before adding the TAT GFP, you dramatically impact the ability to deliver the TAT GFP inside of the cell. So you'll notice a reduction in the overall GFP levels here between this and that. And now the GFP that is residual is more granular. It doesn't get into the cytosol as well. It doesn't get into the nucleus as well. So the functional delivery of TAT is impacted and controlled by the surface RNA, by the GlycoRNAs and the RBPs in these clusters. So this is the way we've been thinking about it. This is the model again. And just to sort of summarize, I hope these are the points that people can take away from the talk today. So when you think about the cell surface and glycobiology in general, GlycoRNAs are a bona fide hybrid biopolymer that are presented on the cell surface. The diversity of GlycoRNAs is enormous, and we're just at the very beginning. There are N-glycans attached to ACE3PU. There are O-glycans that we don't understand yet very much, but they're the major form of glycoRNA. And then more broadly on the cell surface, there are these RNA binding proteins that are participating in binding events and tethering events with the glycoRNAs. They form these clusters, and very critically, they're functional. The RNA and RBPs form these clusters, which are the sites for internalization of things like cell-penetrating peptides. And not only are they the sites for those internalization events, they control the internalization event. If you have or don't have the RNA, you will get different delivery. And we think this is sort of the first example of an RNA-dependent internalization system where RNA can gate what is coming into the cell from the outside. So I'll end there for today and just thank some of the people in the lab. So Jonathan, Peyuan, Christopher, Jennifer, and Shar. People sort of directly in the lab been working on many of these different projects. Of course, our collaborators have been incredibly helpful. Axe is on the call, and just really just want to say again, like this whole set of discoveries around the glycolinker and the glycoRNA would not have been possible without Axe's dedication and skill on the mass spectrometer. Want to thank the funding agencies that have been supporting this, and I'm happy to take any questions, and thanks everyone for their attention.
Okay. Oh, really nice presentation and a lot of work presented. So right now we are taking questions. So.
Hello.
Yep. Hi.
Okay.
Hi, Rain. That's really great talk. I learned a lot. Maybe it's stupid questions that I'm wondering why you only focus on the glyco-modified RNA on the surface. Is that possible that the modified RNA also inside of the cell?
No, it's a good question. All of the original work we did from the cell lysate, so just the whole cell actually. And what we discovered was that, as best as we could detect, there's no sialylated glycoRNAs inside the cell. So the particularity there, right, is that sialic acid is a very late stage glycan modification, and things that get sialylated are usually quickly shuttled to the surface of the cell. So the tools that we have to study glycoRNAs study often the very mature forms. And if you do a measurement, it's like 99.99999% of the sialylated glycoRNA are on the surface. Now, we do think, and we have evidence that there is glycoRNA in the ER and in the Golgi, but they're less mature forms and they're harder for us to study for the moment. We have looked in the cytosol and the nucleus, and we don't see, with our current tools, someone else can prove me wrong later, but we don't see with our current tools glycoRNAs in the nucleus or cytosol. But we think that the bulk of these glycoRNAs are being put on the surface of the cell. Yeah.
Okay, I see. So do you think that the modified RNA are more stable than the unmodified RNA? Because you mentioned that there are a lot of RNAs on inside or outside of the cell, and also I'm wondering, do you have a speculation on the glycan modified RNA's function?
Yeah, absolutely. So we definitely think so. First of all, like you were saying, the outside of the cell is sort of harsh to RNA. There's lots of RNases and many other problems. The small non-coding RNAs are very structured, which makes them more stable. From our mass spectrometry, we see that there are many other PTMs, 2'-methylation, base methylations, base modifications that will add to the stability of the RNAs when they're outside. And then when we look at where the glycans are in the structure of the RNAs. The glycans are positioned on the single-stranded loops of the structure, which would further block ribonuclease activity to destroy them. So we think that the glycoRNAs are sort of the most stable you could generate because they're heavily modified, heavily structured, and then even in the single-stranded regions have glycans to block them. So that's the way we think that they're able to survive in that extracellular environment.
Okay, I see. Do you think that the modification also have impact on the, for example, when you do RNA sequencing, you have to reverse transcribe the RNA. And do you think that also have an impact on the reverse transcription?
Yeah, absolutely. So I think we don't yet have a perfect strategy for sequencing, but we do try to let the cDNA synthesis go for a long time. We use higher temperatures. We use the sort of highly robust RT enzymes to try to get through as much as we can. Sometimes we see truncations, which could be at modification stops, but we really try to let it go as far as we can. But I think it's not perfect. It definitely, the modifications, both the standard modifications as well as the glyco modifications, I think impact the cDNA synthesis for sure.
Okay, great. Thank you very much.
Of course.
Hello.
Hi.
Hello. Thanks for a great talk. And for TAT CPP can also go into bacterial cell. So do you think this glyco-RNA can be exist in bacteria?
That's a very good question. The short answer is that I don't know. You know, the interesting thing I would say is that all life has RNA. All life has glycans. So it's a very curious thing to ask this question. The challenge for us has been that while all life has glycans, the monosaccharides that generate glycans in bacteria and archaea and eukaryotes, they're all very different. So we have developed tools to study mammalian glycoRNAs and mammalian carbohydrates. But the bacterial sugars, the archaeal sugars, even the yeast sugars, they're all very different. So we need different chemical tools. Even the mass spectrometry, the processing is very different. The digestive enzymes have to be different. The masses are different. So there's a lot of complexity moving across kingdoms of life. So I would hope, or I would say it'd be very cool if there are glycoRNAs elsewhere, but we haven't been able to make those measurements as easily because the skill and the knowledge to move to even yeast from mammals is very difficult. So I'd be happy to work with someone to try that, but it's been slow because I don't know anything about the bacterial, for example, the bacteria environment or the bacterial glycans.
Thank you very much for your answer. And for RNA glycan, do you think will affect the RNA binding or RNA function? So what's the main function about what's the main function for its glycan?
Also a very, very good question. I think that there's a primary activity of the glycosylation, which would be to stabilize the RNA. So to make it more stable in the extracellular space. But then once you're stabilized, I think now you're just a new surface. You're a hybrid biopolymer that's outside, and you could interact with other things. You could interact with receptors. You could be a signaling molecule, or you could be an adhesion molecule or whatever. So I think that the glycan probably first enabled the glycoRNAs to exist outside of the cell. But then over time, over evolutionary time, they probably form new functions, right? Maybe they're there to regulate the entry of these CPPs. There might be some other receptor ligand interaction that we're not knowing yet that they're playing a role in. So I think there's multiple reasons that the glycan could impart a function on the RNA.
Thank you very much.
Let's talk. I have a question. How many RNAs with the ACP3U modification that do not have a glycan? So that means can we use the RNA modification ACP3U to indicate whether there's a glycan?
So yeah, ACP3U is interesting. It has been known for quite a long time. The discovery of ACP3U is quite old, actually 40 years ago or something, but no one knew why we make it. So the complexity is that there are cytosolic tRNA with ACP3U, which we don't know the function. They may do something, no one knows. We think, though, if they get into the ER, they can get glycosylated. So when you're secreted, we think that the ACP3U is very important for glycans. But we don't have a way to disentangle a cytosolic role from a secretory role. So the cells were happy, the cancer cells we used were still okay. After we knocked out DTWD2, they were fine. So maybe there isn't an essential role for ACP3U, and we could start to study the effect of the loss of ACP3U and the loss of glycoRNA from that. But you are getting rid of it from cytosolic RNAs. So it's not a clean knockout, if that makes sense.
Okay, thank you.
Yeah.
Hi, Professor Flynn. Can you hear me?
Yeah.
Big applause from me, and really inspiring work. And as you illustrated in the two new research, the cell surface glycoRNA are mainly tRNA, as illustrated in the Cell paper. But in your 2021 Cell paper, you mainly focus on the yRNA and other small RNA. So I'm a little bit confused whether that kind of other small RNA will show on the cell surface.
Yeah, no, it's a great question. So it's a little bit limited or focused on what we could say confidently. So AC3PU has been really well reported only in tRNA. So we focused on AC3PU for the tRNA. But in our sequencing, we saw tRNA and also yRNA, sncRNA, and sncRNA. What's very interesting is that when you look at the sequencing data, and I can tell you a short funny story, but when we first put the GlycoRNA story on bioRxiv 2019, I included an analysis of the stop sites, which nucleotide did the cDNAs truncate at. If you look at the analysis in the bioRxiv on yRNA, on sncRNA, on sncRNA, every single time the cDNA truncates, it's at a G. Okay? It's different on the tRNAs, but on the yRNA, sncRNA, and sncRNAs, it's always at a G. When we went to Cell, they said that's not validated enough, so they didn't want me to include it. So it's actually not in the Cell paper. Now you come back to the new work where we did the mass spec. What did we find? We found two bases. We found uracil through ACE3PU, and we found G, guanosine, through Y-butysine, the hypermodified G, Y-butysine. So my prediction is that the linkers on tRNA are through ACE3PU, as we showed, and that's through the uracil. But the linkers for yRNA, sncRNA, and sncRNAs are through this hypermodified Y-butysine structure, because it's exactly where we would expect in the sequence. It's on a G, and it's these other RNAs. So we ended up focusing very specifically on the ACE3PU for a chemical synthesis perspective and the specificity there in the mass spec. But I think this is the way it's going to work out, is that there's going to be two major linkers for N-glycosylation, ACE3PU and Y-butysine. On the tRNAs, you get ACE3PU, and on the other small encoding RNAs like yRNAs, you get Y-butysines. And this is where they're going to be modified on the Gs. So I think they're both there. We know they're both there, but the way the mass spec was working and the way we could do the synthetic standards was very limited on the Y-butysine side of things. So that's the way we ended up moving those things forward.
Well, thank you. That almost answered my second question, because I was wondering whether the Y-butysine is the other substrate for the other small RNAs. And another small question that confused me is that you showed that the glycoRNA and the glycoprotein, they share a similar or almost the same pathway. So I was wondering whether there is a specific enzyme which catalyzes the installation of glycan on RNA, GlycoRNAs.
Yeah, for sure. So we don't know. I would say we're still trying to do some of this very basic discovery, but what I can tell you is when you inhibit N-glycosylation with a small molecule, so you inhibit both OSTA and OSTB, you get a reduction in the glycoRNA. And so we think that there is a common glycosyltransferase, and we think that the glycosyltransferases are the same as what happens for proteins. So we think there's a shared mechanism between the RNA glycosylation and the protein glycosylation. So that is the strategy we think that the RNA takes to get glycosylated. So we don't think there's a special RNA glycosyltransferase, but we think it's shared between the protein pathway and the RNA pathway.
Well, thank you. Thank you very much.
Hello, Professor.
Hi.
Hello. Thank you for your presentation. Professor, I have a question regarding the molecular weight or size of the glycoRNA. As I have read your paper, and among all the Northern blot papers, you have mentioned that their molecular weight is greater than 9 kb. So my question is, you have used downward capillary transfer of RNA by applying Northern blot. So as the molecular weight is so high, so is it possible to transfer these RNA in around 2 hours from the gel to the membrane? And the second question is this: if we see the gel imaging of your total RNA, we were unable to see the image of glycoRNA. So what about the concentration or total amount of glycoRNA you get?
Yeah, so if you want to take a look, the methods are very detailed in both of the papers, but yes, you can do the transfer in about 90 minutes and get full transfer. And then the molecular, the concentration is indeed very low. There's a couple of things to just keep in mind. Small RNAs are harder to detect than long RNAs with cyber gold, cyber safe, whatever. And the diffuse nature of the signal is going to lower the signal to noise. So we do not see cyber signal in the high molecular region because there's low concentration and it's diffuse, it's on a single band. So this is the problem with looking for the cyber signal up there. Just to make a note, I do have to leave in a few minutes, but I saw, I think Chi Chao maybe what had one last question, which I could try to answer.
Hi, Ryan. This is Chichao from the Guangzhou Art Club. So I would like to ask how comprehensive is the glycoRNA binding to the proteins besides the DDX21?
So
Can you hear me?
I would say that we have this is actually, we put it in a preprint, so it's on bioRxiv, but we know that there are dozens, so 20 to 30 RNA binding proteins on the surface of the cell. But there's a lot of heterogeneity that we don't understand yet. We don't know which RNA is bound to which RBP and where they are on the surface of the cell. There were many dots, right? So there's a lot of details that have to be worked out for which RBP is where, and then which RNA is bound to which RBP. So we sort of just have a map and says, these are all the RNAs and these are all the RBPs, but we don't know the pairwise information yet. Yeah.
Okay, thank you.
Great. Yeah.
I guess I will just ask the last question for everyone.
Sure.
So yeah, thanks so much for accepting the great invitation from us and give us the opportunity to talk to us from China. And just want to ask from more general, like the discovery of this glycoRNA is kind of revolutionary for glycobiology and RNA biology. So since you're first bioarchive since 2019, so several important studies has been published. So of course, it also comes with a lot of controversy. So given this, could you just give us your insights on how scientists should, approach this challenging and especially these kind of crazy ideas in biology?
Yeah, that's a good question, X. Yeah, I think it's interesting. I think it depends on the way one is trained and the framework that you like to think about the world and science. And so very early on in my training, when I was an undergraduate at MIT, I learned that I got an experience that told me we know very little about systems that we've studied for a very long time. That was very important for me because what it meant was it's always okay to ask crazy questions. It's always okay to wonder, why wouldn't this happen? Why can't this happen? So when I think about biological problems and questions, I always wonder, why not? Why not this answer? Why not that idea? Now that does open you up to a lot of questions. So you can get a lot of criticisms and these sorts of things. But I think that my strategy has always been get very good at making a measurement. Make sure that you know exactly what you've done in the lab, on the computer, wherever. And then you can report that. And so if you're very confident that you know you made that measurement, you might not be perfectly right. The model you drew might be a little wrong. Someone might find something else out. But the whole idea is to advance all the knowledge as fast as we can. And so, for example, I'm very, very particular when I was writing papers by myself and now as my lab, that all of our methods are very detailed, right? Everyone should be able to read it. And I think, people will email me. I try to get back as soon as I can with exactly what we did. What is the concentration? What was the test here? Whatever. So it's been very interesting because some people are critical but excited to understand, and some people are just critical. And so I think that it's better to be the first type. You can be curious, you can be wondering, you can, think, well, this doesn't make exact sense. Maybe I should try it myself, or, I don't know, this could be interesting, but it's not what I would have thought. But I think if you're it's sort of like a frame of mind. If you are wanting to be negative, well, then this is more boring, I think. And so it's been interesting, like you said, to see, because I didn't start off thinking I wanted to change glycobiology, but it was the results of what I worked on in Carolyn's lab, and then now what my lab is doing. And so the way we think about it is like, we like asking the questions that we think are interesting. We want to share whatever we see, and we're very open. If someone tells us you did it wrong, if someone says there's a new system to try, or you should do this, we try to do it as quickly as possible. And so, yeah, I like to share, I like to share the stories and get people thinking about it. And that's why I started the talk. I think we are very early there. I mean, even 9 months ago, I didn't think a lot about O-glycans, right? And so there's still a lot of work that has to be done, where there's fundamental observations that will, add to the story, make it more complex, make it more specific. And so this is the way I like thinking about it. But yeah, hopefully that makes sense.
Okay, the answer is great, and X after yes. Today it's already like quite a long time, but I thought the time is flew very fast. We learned a lot. And I have to say, like X mentioned, the work actually put on bioarchives more than 2 years before it's finally published on Cell. So like Ryan is famously really likely to share their new discovery on bioarchive. I thought at that time, because at that time bioarchive is not so popular, but then I heard the GlycoRNA stuff on Twitter on bioarchive, or I said bioarchive is great, and I found Ryan's most paper are put on bioarchive, so that's really helpful for the community, for people those things 2 years before the publication. That's really great. Thanks, Ryan. I said, yeah, in future I will do the same. Yeah.
Wonderful. Well, yeah, again, thanks everyone. It's been really nice. And one day I'll be able to come in person, I think, and see everyone.
Yeah, definitely. Hopefully
in Guangzhou, yeah.
Yeah, in Guangzhou.
Awesome. Yeah. Okay.
Okay, thanks again.
Yes. All right. Bye-bye, everyone.
Okay.
Thank you. Bye.
Recording stopped.